Hello, good morning, everybody. Welcome to today's webinar on oil spill incidents and management. I am Gayatri Balan, moderator for today. I represent Barkavi Scientific Events and Marketing Services, which is hosting today's webinar. This webinar is organized by SG Lab Forum, Singapore, and sponsored by Frontier Lab. Today we have two interesting presentations on petrochemical industry related applications. The first presentation focuses on oil spill incidents, characterization and evaluation, while the second presentation is related to petrochemical process evaluation. We have two speakers, Dr. Liu and Dr. Satrugnan, to enlighten us for the next 1.5 hours. The first lecture will be delivered by Dr. Liu. The title of his presentation is Thermal Slicing Ramped Pyrolysis-GCMS, a powerful technique to elucidate composition and structure of oil residues in environments. Let me briefly introduce Professor Liu. Dr. Liu received both BS and MS from Xiamen University, China, and a PhD in coastal oceanography in 2006 from the State University of New York at Stony Brook. He then spent 3 years at Old Dominion University as a postdoctoral research associate. In 2009, he joined UTMSI and is currently an associate professor. Dr. Liu's research focuses on understanding sources, transformation, and fates of natural and anthropogenic organic matter in aquatic environments. Now I request Dr. Liu to deliver his lecture. Also, I'm requesting all audience to post their questions in the chat box. These will be addressed during Q&A session after the lecture. Thank you. Dr. Liu.
Thank you. Thank you for the introduction. I also want to thank the invitation by the SG Labs. So I'm going to talk about some of the work I was involved after the Deepwater Horizon, mostly this pyrolysis technique that you see from the title. Before I go, I do want to acknowledge my lab personnel. You can see some of the names, including postdoc, or visiting student, or my grad student, the lab technician, etc., and some collaboration from different universities, including University of South Florida, and certainly the UT and the Baylor University, etc. The funding mostly from the National Science Foundation, but also the Gulf of Mexico Research Initiative, essentially the BP money. They managed by the Gulf of Mexico Research Initiative, and you can see we're funded under this platform we call the DROPS. I think the slide is frozen. Okay, here you go. So here's the brief outline, what I'm going to talk about today. Certainly briefly talk about my research, what I focus on, and a brief introduction of petroleums, kind of some background information and the Deepwater Horizon oil spill. And then let's go to the story based on some field observation data and some laboratory incubation, and also certainly we'll go to the technique, the pyrolysis technique we call thermal slicing ramp pyrolysis, coupled with the GCMS, and make a conclusion. I define myself as an organic biogeochemist. My research direction mostly to look at how the environmental condition affect organometal decomposition. So I have this simple equation here. That you can see CO2 plus water, right? This ring about like a photosynthesis. You produce this very simplified organic matter form, CH2O. So essentially glucose, right? I just simplify with 1 carbon, 2 hydrogen, 1 oxygen, and you produce oxygen. When you go to the right, you produce organomatter by the algae, by the plant, but you go to the left, we call the organometal decomposition, right? You go back to CO2, you go back to the seawater. So in nature, you always have these two competing process. You know, you produce organomatter and then you break down the organomatter. In a sense, this is like the Chinese philosophy yin and yang, right? You have the yin and yang competition. And this kind of a little bit complicated cartoon to show you, you know, we deal with all type of organomatter from the fresh biota, algae, zooplankton, etc., and also non-living organomatter. Probably you guys heard about the geopolymer, humics, and this go down further decomposition, you form the keratin petroleum, right? They take a long time scale. But eventually all the organomatter will go back to the CO2, right? Like the equation I have here and the nutrients. But this process will be determined by the environment condition. It determines whether you have bacteria, whether you have the sunlight, whether you have enough nutrients for the bacteria to break down the organomatter. And certainly now we have the plastics, right? Plastics is also a part of the organomatter in the environment that they can be decomposed. So my research involved all type of organomatter, either the particular organomatter, either suspended or sinking particles in the ocean, and the dissolved organomatter in the ocean, small peptide, proteins, and the petroleum hydrocarbon. I also do a lot of microplastic, but today I will focus on the petroleum hydrocarbon. I use, because I do the organic compound analysis, I use a lot of instrumentation. So you can see the instrument in my lab. I got a lot of this LC liquid chromatography coupled with different detectors, including mass spectrometry. And I have the pyrolysis. This is one of the main techniques I'm going to talk about today. And have some, you know, gas chromatography, mass spectrometry, and so on and so forth. So I rely a lot of this kind of advanced instrumentation to quantify the organic compound in the environment sample. So let me give you a brief background. What is really the petroleum, right? It's really a complex mixture ranging from the methane, or the CH4, really simple form, and all the way to asphalt. You know, this is the stuff we're using to pave the road. And they can have, you know, gases, the liquid, condensate. We often say petroleum, sometimes we say crude oil. Crude oil really refers to the liquid phase of the petroleum in the atmosphere pressure. You know, if the crude oil, the gas will be gone, right? So, but the petroleum including both gas and the crude oil. And you also have some trace compounds like trace metals or nitrogen, sulfur, etc. Right? So if you look at this right-hand cartoon, this shows you the crude oil after distillation. Right? We have to process the crude oil in order to use them. For example, this x-axis here, based on the carbon number, you have 4 carbon to 10 carbon. We define the gasoline, and then you go to the right, the kerosene, diesel, and heavy gas oil, lubricating oil, and all the way to the asphalt. Right? You can see as the carbon increase, this kind of become heavier and heavier. And if you look at the component, the compound here, the y-axis, is look at show you the composition. So the gasoline dominated by the N-alkane, we call it normal paraffin. So you got 80–90% of it. You go to the right, you can see the amount of the aromatic fraction increase. In other words, if you have the L14, there are a lot of aromatic fraction in this range, right? So that gave you a simple idea of the composition of the crude oil, right? But here's another way to see it. You know, the crude oil, what is essentially the structure or the compound we say about, depending on where the crude oil from, you have 75% aliphatic hydrocarbon. They're very simple molecules. You can see, you know, every Vigo is 1 carbon, right? It's saturated by the hydrogen. And those compounds can be straight chain or iso chain, right? Or sometimes cyclic. But either way, they are saturated. And this group of compounds, we often call the saturated hydrocarbon. They are relatively non-toxic, right? It's not really a concern when this compound leaked into the environment. And then you have about 15, 20% we call the aromatic And you can see they can have some aliphatic tail, but essentially the benzene ring, right? You connected them together. This unsaturated hydrocarbon, they are highly toxic. This is really one of the components people concerned about when you have an oil spill, right? They're toxic. And then you have a very small fraction of the racine, we call the L14. They are structurally very complex, and they are a little bit polar. Because they contain sulfur, oxygen, nitrogen, etc. And because they're not really easily soluble, we often say this is a little bit low toxicity. So that's kind of give you an idea of the chemical composition. As a chemist, or if you try to evaluate the toxicity of environmental sample, you have to do the compound analysis, right? So typically we call the traditional chemical analysis. We often say, you take a sample from the field when the oil spill occurred, and you analyze them. How you do it? You take a sample, you're using this Socklix extraction, right? You put the sample in this little vial here, and you have the organic solvent that you boil, it kind of extract all the organic compound, hydrocarbon into the solvent. And then often you do a column. So this is a chromatography column, and you can fraction them. You can have saturated fraction or aromatic fraction. And then you collect the different fraction, and you do the analysis, either using gas chromatography, flame ionization detector. Often we're using this instrument to do the alkane analysis, or we're using the gas chromatography mass spectrometry, or the GCMS. Often we're using to quantify the polycyclic aromatic hydrocarbon or alkylated aromatic hydrocarbon. This is kind of the people often used to do the oil analysis. But the problem with this technique, you only have a limited analytical window. In other words, often you can only quantify about 25% of the whole crude oil. So it's very limited. So there's the need to expand the window, of course. And the other way traditionally we're using, we call the cellular fractionation. So essentially give you a crude oil sample, you using different type of organic solvent based on the solubility. For example, you using DCM, dichloromethane, to extract the environmental oil sample, and then you add some N-heptane, which is the 7-carbon saturated alkane, and anything insoluble we define as 14. And you can see when you change the organic solvent, you can, anything soluble, and hexane we call saturate, or toluene here we call aromatic. So this way you can simply weight, right? So you can get a really excellent idea how each fraction distributed, because you can use the balance to weigh them, right? So it's an excellent quantitative way to analyze the sample, but you don't have the molecule level information. You don't know the organic compound, but you do know the mass, right? How that fraction will change for different crude oil, or in environment for a longer time, so you can quantify how the relative fraction will evolve in the environment. And this kind of give you a picture. You can see the saturated, you don't have color. Aromatic, because the aromatic rings, they have a really dark brownish color, and you have the L14 is almost a solid phase at the end. So let me briefly talk about the deepwater Horizon oil spill. This occurred in April 2010, and it occurred about maybe you guys. This is the Gulf of Mexico. This is Louisiana. This is kind of where the Mississippi River kind of entered the ocean. So about 50 miles off, this is the city of New Orleans. So about 50 miles offshore, this is where the oil spill occurred. You can see this picture is a wellhead in the deep ocean, and some oil leaked to the sea surface. They flow on the sea surface because the oil is lighter, right? It's a little bit lighter than the seawater, so they tend to flow on the sea surface. So the responder, they using the in-situ burning to get rid of the oil. And certainly a lot of oil, because of the ocean current, they push the seashore. This is kind of red brown color, which is suggesting the contamination by the oil. You can see the picture in the marsh environment. There are a lot of brown, dark oil get pushed to the shore, and the seabird. And the amount of oil, about 200 million gallon of the oil. So this kind of put all the previous historic oil spill. This is really one of the largest oil spill ever occurred. And maybe you guys remember there are other names here. For example, the Exxon Valdez in Alaska. This is also very famous oil spill. And the bottom bar, the kind of put you a perspective how much oil we're talking about. Global consumption, is about 150 million gallons. So that Deepwater Horizon oil spill leaked more than the whole global consumption of the oil. So the point is there's a lot of oil being leaked to the Gulf of Mexico. This oil spill is a little bit unique in the sense because it's the first time the oil leaked from the deep ocean. So deep ocean, I mean, is a 1,500 meter, right? This kind of the cartoon show you where the wellhead being exploded and the oil goes through the water column. To me, because I mentioned I'm interested in figuring out how the environmental condition affecting the oil degradation or the weathering. If the oil rises from the deep sea floor to the sea surface, as you can imagine, they can subject to a large environmental condition change, right? In the deep ocean, in the dark, high pressure and the low temperature, but in the sea surface, now you have the sunlight, maybe you have the photo oxidation, and you have a high temperature now, and the nutrient is depleted. The deep ocean, we know there are a lot of nutrients. So this environmental condition changed a lot. So the idea they may change, how the oil being degraded. So it's kind of provide a fascinating scenario to study the oil degradation. I think this right hand kind of cartoon show you the oil composition. Sorry, this is probably not easy to read, but simply the x-axis show you from the leftmost side, the methane, all the way to the C40, so the carbon number increase. So this line, this green line is to show you the whole crude oil composition, but this is different color, which means if you take sample from different places, you get a totally different oil composition because of the oil, when they rise to the sea surface, there are a lot of fractionation. Because of the solubility, because of the evaporation. In the air, you got a lot of this low molecule weight hydrocarbon. They are more really volatile. But in the sea surface, the oil slick, this is what you got. It's very heavy or higher carbon number, the hydrocarbon. But in the deep water plume, this red bar, that's what type of the hydrocarbon you got. A lot of small aromatic compound. So I got involved in the research. This is kind of, this is a picture I took during the oil spill. We had a cruise to survey the oil spill. You can see it's a lot of pictures, different oil mousse, and the different color, which is suggesting the subject to the different degree of weathering. And here the marsh, here the sediment we took, the sediment core tube. So my initial research question is how the environmental condition affecting the oil degradation, right? I think that's important question for the policymaker, for the community, because only know that you can predict the fate of the oil, how fast they can last in the environment. The only quantitatively get the idea, then you can predict how much oil left in a month or a year or 10 years time scale, right? I think that's important data we need to have. So the first data we got, we published this paper in 2012. So to look at I have a lot of data, but I will focus on the PAH here, the polycyclic aromatic hydrocarbon. As you can see, okay, here you can see the kind of the EPA, the Environmental Protection Agency of the US, defined this 16 PAH is a priority pollutant. So it's really concerned in terms of toxicity. So we measure this 16 pH in different spot of the oil. This figure is a satellite image because of the oil has, the color, so you can see it from the satellite. Here, the oil spill side, you can see the kind of the globs of oil floating on the sea surface. Because of the ocean current, they try to push the oil away, right, to flow to the west a little bit and go to the east, and the so-called loop current. So you can see it takes time for the oil really freshly leaked and go to this oil slick side and the CT side and the marsh point, the kind of the shore. So there is the time sequence. So based on those samples, we measure those total concentration of the pH. You can see the pH actually decreased more than 90%. So that pH are gone mostly when they hit the shore. So which is a good news actually. Why the aromatic hydrocarbon gone, we don't know, right? That's interesting. But it actually is a good news because of the mean the less toxic. But the question is what happened to the pH? Why they're gone, right? Why they decrease? And you're talking about maybe a couple of weeks here, maybe a week or 10 days from the DWH side to the shore. So that means the pH being weathered or being degraded really fast. We know the degradation involved for the aromatic hydrocarbon to be degraded. Maybe you have the photo oxidation because of the sunlight, the oil there on the sea surface, or you can have biodegradation. We know a lot of bacteria, they actually like to chop up the oil, right? This is kind of the cartoon show you how the photo oxidation works. So you form a lot of radical, and those radicals break down the compound, the aromatic hydrocarbon. And we design an experiment to try to evaluate why the pH is gone so fast. It's because of the photo oxidation, because of degradation. We did this is my postdoc. We did some on-deck incubation to kind of elucidate, to differentiate what the role of the sunlight, what the role of the biodegradation. We got a lot of treatment, right? So you can only do this by the incubation, because you can control the condition. You can say, okay, this sample with light, without light, that sample with bacteria, without the bacteria. So you can manipulate the treatment. You can get an answer, right? So in that sense, we have a dark control in the dark, and we have without the bacteria, we get rid of all the bacteria. So there's no biodegradation. And we have the bacteria in the dark oil, but without the light, and so on and so forth. We also try the dispersion as well, and the light treatment will cause to expose all the sunlight. Here the setup we have. All the bottle is quartz. Quartz, we don't use typical glass, we use quartz because quartz allow the UV to penetrate. Let me show you some quick data, simple data, right? This is kind of the time, how long the experiment lasts, about a month. This we normalize the initial oil concentration. We kind of are using the total concentration of the pH. The time zero is 100%, right? With time they being degraded, they go down. So this is a black curve here, the black square. This is the control. This is no light, no bacteria. This two purple is dark. It's dark with bacteria, but without the light. So you do see they decrease. I mean, pretty fast actually, but you have a 10% left at the end of the incubation. These three dashed lines, all the light treatment, whether you have the bacteria or not, whether you add a dispersion or not, they about the same, they converged. I think this data clearly show the photo oxidation, the light, play a key role to destroy those toxic aromatic hydrocarbons. I think that's really clear message, okay? And then you can just, there are a lot of individual compound data you can do. You can assume a first order reaction. You can calculate the reaction rate, read constant K. This show you each individual pH, which one is the most light sensitive. So you can see here, naphthalene here is really photosensitive, and racing too. So different pH, they have different light sensitivity, which is expected. And this right-hand figure show you the model, what the role of the light versus the bacteria. You can see there are some, the black bar show you that actually the longer, which suggesting they are more easily to be biodegraded. So anyway, I won't get into detail of this, but I'm trying to say the light is the key here to destroy the pH. And this gives you an idea. We call the light photo oxidation. Some people say photo bleaching in the same sense, right? You can see this is kind of after the oil we extracted with the DCM, the organic solvent. You can see after 36 days, whenever you have the light treatment, this is the 3 light treatment. After a month, the color gone. So clearly another sign of the aromatic hydrocarbon being destroyed. And how they destroyed, we got a lot of this kind of hypostatic pathway. You can see you have it's a model compound that enters in 3 ring pH. After the photo oxidation, you can see you break down the ring, you get a lot of oxygen added in. When you break down the ring, I think that's explain why the color is gone, right? I won't be able to get into detail of this, but this is essentially show you the possible pathway, what happened when you have a light to destroy the hydrocarbon, the aromatic hydrocarbon. The question I'm interested further, they being destroyed, then it's not like they go to the CO2, right? They can't go all the way back to the CO2 unless you're talking about biodegradation. So maybe there are a lot of compounds being formed. So I'm interested to see what exactly, how the pH being transformed to what. So we designed the experiment to look at the fate of the oil after the photo oxidation. So this is my visiting scholar, Qing Wang. So we did the experiment. You can see this is from the lab. We have the cross bottle, but this time we focus on the L14 because that's what being formed after the photo oxidation. So we're trying to interested to look at the how exactly the oil changing, you know, after the photo irradiation or the photo oxidation. Again, as I said, we're using the Cellular. So the first step we do this to separate the oil to a different fraction. We're using this Cellular Fractionation. So you can see here that this is about again the 1 month time scale. This is a different fraction. You have the saturated fraction, all the solid line or the treatment, the dashed line or the control or the dark. So the no light exposure. So the saturated hydrocarbon, the N-alkane or the cycloalkane, they don't change much, right? You can look at the weight percentage, about 60%, they don't change with time. And the aromatic, see, it started from about 25%, but they decrease about 50%. And you have the racine, they don't change much. Look at the L14, you get about 0% L14. I mean, this crude oil is a Louisiana light sweet. It's really good oil, which means you don't have a lot of L14. But look at the light treatment, you increase that dramatically. You know, 5 days, you already have from 0 all the way to 10%. So the other way to show the data, you know, I kind of using only the day 44, the end point. You can see how the relative percentage change, right? This you only take the time 0 data and the time 44 data. Here to show you the cellular fractionations, how they weight change. You can see the saturated, you decrease about 3%. You probably can ignore that. But the aromatic, you know, 25% all the way decrease to 13%. 50% decrease. And that fraction proportionally changed the L14. So you got about 1%, but you increase to 13%, which means because of the photo oxidation, again, this experiment, there are no bacteria or autoclaved seawater, so the purely sunlight. So you got a tremendous amount of L14 formed after a month. But that formation is really fast, actually. If you look at the weight change, about 10 days, you already get like 10% or so, right? So that means the process is really fast, and the aromatic change to the L14. So our research question turned to, what exactly the structure of the L14, or we call the photo-generated L14. How we do that? That's actually not easy, right? You can't using the GCFID or the GCMS I'm talking about, because they're not really that soluble, because the structure is complex. People using this, we call it FTICR. I mean, this is really a fancy mass spectrometry, but this not quantitative, and the sample has to be really soluble. After the oil spill, there are a lot of tall ball form. They're not really easy to be soluble. And people can using the nuclear magnetic resonance, we call it NMR, often based on the C13, or the isotope C13 carbon. They do the solid phase, and they're very quantitative, but it's only a function group. So there is no molecule level information. So then that's one of the reasons I turned to the pyrolysis. Some brief knowledge about the pyrolysis. Pyrolysis is simply means thermal decomposition. So you gather some organic material, you heat it without oxygen. So you don't burn it, right? You heat it, so you thermally decompose the material to the smaller fragment, and then you can analyze any fragment coming out. We call pyrolyzate. People often call it pyrolyzate. And this technique of pyrolysis is old. I mean, maybe century old. People have been using this technique a long time. I mean, the typical example of the we call it rock evolve pyrolysis is people who studied petroleum or the kerogen. They often using this technique, heat the sample, the rock sample, to see the potentially how much oil being generated. You can see this is a temperature program, and this is the kind of the fragment being released. You can monitor by GCFID, for example, the different fraction. You can see maybe the first fraction is very volatile, to show you the potential, right? And then often combined with some people using CO2 analyzer, and so on and so forth. So pyrolysis is a really old technique. And one of the popular one to combine with the GCMS, we call the Curie point, which means you heat the sample at a certain temperature, such as 500 Celsius degree, 600 Celsius degree, maybe a couple seconds, and then you can monitor any compound released. But there's a drawback, right? You don't know for a given compound, you don't know, whether they're at what temperature they can be released. But any compound released at that temperature, I think it don't give you really clean information. I think the other technique they call the multiple code trap. What they do essentially, they design this multiple trap, and they pyrolyze the sample at a certain temperature, and they condense each individual trap. In other words, each code trap represents a certain pyrolysis thermal slice you chose. You say, okay, 100 Celsius degree to 200, I'm going to condense the pyrolysate in this trap, and 200 to 300, the second one, so on and so forth. This is a paper published, I think it's 94, by Tang and Stouffer, but this is the kind of the lab homemade instrument, never really popularized. I think it's a little bit complicated to use because of the way you have to do the multiple trap. And this is where it kicks in the thermal slicing. And this is really the frontier technology. They call it hard cut. I name it thermal slicing, but I think it's easy to be accepted by the scientific community, because thermal slicing is easy to understand than the hard cut. But anyway, this technique is simple, right? If you let me walk you through. If you have some type of solid sample, you don't have to do any pre-treatment. You simply just freeze dry the sample, which is really, really easy. If you have oil sample in the environment, you take it solid. You don't have to do the organic solvent extraction, all the tedious procedure. All you need is to take a really small amount of sample. I show you here 0.1 milligram, and you drop the tiny cup on the top, and then you go through this pyrolyzer, and then you pyrolyze it using whatever temperature you choose, and say, okay, 50 to 90 Celsius degree, you heat it, and any compound coming out of that slice will condense, we'll call it a cryo trap or the cold trap. This trap connected to the liquid nitrogen. So any compound basically will condense here. And then you stop at a certain time interval. Okay, I got enough of the pyrolyzate, so I can stop. I can just disconnect this, disconnected this liquid nitrogen, and then you can program your GC to run through the column and run through the mass spectrometry, right? And then you get a chromatograph here. And then you can just increase your temperature slice 90 to 170 degree, and so on and so forth. So essentially, you get the two-dimensional data, right? So not only the GCMS, but also you get the idea at what compound, at what temperature they can come out. So I often call this the peel the onion technology, right? When you peel the onion, one layer by the layer, you can imagine one layer represent a thermal slice at what temperature you want to do it. So this I gave you example of the two-dimensional data, which x-axis is the retention time with the ion intensity, right? This is the standard GC mass spectrometry spectrum, right? You have the retention time, you have the individual compound, but this you have a series of thermal slides. You essentially have the two-dimensional data. When I first used this technology, I was really pleasantly surprised how well this works with oil sample, because we can easily separate the slices based on how the compound is coming out. The 50 to 370 Celsius degree we call the thermal desorption zone. Why we call the thermal desorption? Because at this temperature range, we don't think there are a lot of cracking happen, because when you heat the sample, often you charring the sample, you cracking, there are a lot of compound. It's not the kind of, how to say, the original compound. You artificially produce the compound, right? That's one of the things people sometimes worried when you do the organic geochemical analysis. But on this temperature range, I don't think we need to worry about it for the oil sample, because it's a clearly separation. This range is all intact hydrocarbon. So originally the sample had this type of compound. And then you have the cracking zones. It's above 370, which means now you start to break down the covalent bonding. So you generate a lot of fragments. So we have a lot of sign of that, because, for example, you have the doublet here. You can see if alkene, alkane. So it's alkene, alkane. They have the same chance. If you break down a alkyl tail, it's the same chance you form alkene versus the alkane. So that's why you have the doublet. That's really clear sign of cracking. But here you don't see those doublets among all the evidence. But anyway, so what I'm trying to say, it's really work beautifully for the oil sample. So how you do with the data? So one thing you can do, you can see at what temperature this compound coming out. So you can normalize 100%. For example, the C18 alkane, the crude oil, right? You can say, well, you only have 10% coming out of 50 to 90 Celsius degree. The majority came out of this blue is 90 to 170 degree, right? And you have a really small fraction to 170 to 90. But when you go to the right, you increase the carbon chain, certainly the temperature you need to become, for them to become a little bit more volatile, you need a higher temperature, right? C7, for example, C37, now you get really using this high temperature, 170 to 90 degrees. So this is really have to be consistent, or, as expected, to their boiling point, okay? So it's really depend on the carbon chain. So how we do with this data, if you compare, this is a crude oil. So if you compare the oil sample you collected from the field, which is subject to weathering, different degree of weathering, certainly 46 days here is, about 2 years. And this is highly weathered, right? This is the line, the crude oil line. So I kind of fixed the line here. So this is the same crude oil. And you can see any given compound, if you have, they stay in the environment longer. Now for the same compound, if you let them coming out of that matrix, and now you need a higher temperature. For example, this is C20 right here, right? So you got about 70% right here, but the C20 right here, now you really need this purple, the pink color, the 170 to 90 degrees. So it's a lot more coming out, use a higher temperature. In other words, the same compound, if they stay in the matrix become a lot tighter because of the weathering. So you need a higher temperature for them to be released. In other words, the binding energy is getting higher. I think this is the only technique in the world for you to get an idea of the matrix effect. We know that when the tall ball formed, oil spill occurred, you eventually form the tall ball. There are a lot of intact hydrocarbons. This means this provides a way to see how tightly the intact hydrocarbon being buried into the matrix. You actually can go a step further because at what temperature that compound being released, you can calculate the activation energy. So in other words, you can actually give a really good number, or how many joules of the energy you need in order to release that compound, or you can quantify the matrix effect. So this is a paper published by Hemingway. It's also the pyrolysis, but they're using the different technique. They're using the CO2. They add a little bit of oxygen. So all the organometal being burned to CO2. This is a color. This show you the CO2 coming out when they increase the temperature. Because of the temperature, Hemingway et al. Writing a code using Python code, they can calculate the activation energy. So that's what they did right here. So you can see the energy. So you can quantify the energy. I think that's wonderful, right? In a sense, you actually can quantify the matrix. Okay. So that's kind of the tool we have. So what do we do? Back to the question, we want to kind of generate an idea. What the I think I'm doing good with time. So I try to get an idea the photo generate L14. What exactly the structure? So we have this is L14. I showed you this figure before. So we collected this sample, the L14 sample. We run through. This is the pyrolyzer I have here. This is a frontier on the top of the Shimazu GCMS. I'm using this TRSRP GCMS. I also collaborated with Dr. Bill Hogday at the Baylor University using the NMR, which is very quantitative, although you don't have the molecular lab information, but you do have the function group in terms of the information. So let me show you some of the data. I think this is kind of the peel the onion analogy. Okay, so this is a day 0. This is kind of the original L14 in the crude oil sample. So we did. So we pyrolyzed the L14, the original L14 with the different temperature slices, 90 to 170 degree, and all the way to 530 to 650, right? And this is a compound. The left side panel show you the small aromatic hydrocompound from the naphthalene all the way to the C1 chrysene, and this kind of the y-axis show you the different temperature slices. You can see the blue, the green bar here, the low temperature slices, right? And the right hand is an alkane. You can see how nicely you can peel the onion, right? To see, wow, there are a lot of intact hydrocarbon in the original L14. And remember, this is a escape because we're using the N-heptane to precipitate the L14. So which means they're still in the L14 matrix, which means that intact alkane actually escape organic solvent extraction, right? So if you do the classic GC analysis or the extraction, you can miss this fraction because this only you can get those by heating them from 90 to 170 degrees. In other words, this energy, the heating energy, I think is a little bit stronger than the organic solvent extraction. But anyway, this is the kind of original oil. So this is the dark. So 44 days. This is a crude oil in the water, and 44 days, and you do the L14. you can see amazingly how similar they are. In the dark, there's no light, of course they're similar. But it's also suggesting the L14 structure is pretty stable, even 44 days in the seawater, right? But if you look at the light, totally changed, right? I think you see how much stuff you added at the high temperature slices, you know, especially the yellow here, 370 to 530. Not only the small aromatic added, but also this. Look at this, NLK, right? Again, this is a cracking zone. I think I talked about that. This is not intact. This is the breakdown product. But regardless, this is suggesting there are a lot of this compound being added or photo-generated L14. Okay, and this also a little bit increased too. But amazingly, this green bar still have the same distribution. I think that's very surprising, suggesting that's really important part of the structure of the L14. Even though there are a lot of photo oxidation going on, but you still maintain. Again, this is intact. This is really low temperature. This is no chemical bond involved, but I think it is bonding tightly. This is the important part of the L14 structure. Using those calculation I show you with the Hemingway inverse master, the Python code, we determine it's about 100 kilojoule mole. So that's in the range of the if you guys know the hydrogen bonding, that's kind of the energy range. I think this is really exciting data to be able to quantify, how many, there are a lot of intact alkane is important part of the structure, not only the original oil, but also the photo-generated L14, not only the original L14. So you can see how powerful this thermal slicing technology is. I just want to mention point. This is kind of how we do the cellular fractionation. You can see they have the using the N-heptane to get the L14 out. So which means those intact hydrocarbon we found it's really escape this N-heptane. In other words, they don't dissolve in heptane. They have to be kind of not a chemical bonding, but I think it's where the intercellular or intermolecule energy kind of bound into the L14 structure. And we're using the NMR I think I want to quickly show you this data. This is a C13 technique. We're using we can look at the function group. There's no molecule level information, but we can know the function group distribution. So this is we're using this ppm. This is kind of the frequency here. This represents 100% of the carbon we can see. So each peak represents a different carbon function group. For example, this 0 to 60 ppm represent the aliphatic carbon, and this, about 60 to 70, this is COH. Often you see the carbohydrate, for example, this aromatic carbon is big peak. So the oil, if you see the day one, day 0, this L14 certainly dominate the aliphatic and aromatic. But I want to draw your attention later in the incubation, after the photo irradiation, you can see how much oxygen being added in, right? Now you don't have the COH group here. Now when you show up, and you don't have the COO group, you don't have the CO group. Now you get a lot of oxygen being added. So that's clearly show you the photo oxidation actually add a lot of oxygen atom. I think this figure we further, because of the different function group, this is still based on the NMR data to break them down to further different function group, alkyl carbon, N-alkyl, or methoxy carbon, etc., etc. We be able to quantitatively show how the different function group change in the different cellular fractionation after photo oxidation. So I don't think I have time to go through all of this detail. But at the end of the day, we take advantage of all the data, including the thermal slicing pyrolysis, also the NMR. We come up with this molecule model is really hypothetical. but this will show you how the aromatic hydrocarbon changed to the photo-generated L14 based on the cellular fractionation data I show you. So this we think is what happened. This is kind of hypothetical. It's a kind of average molecule, right? You got a small aromatic, but you got this long chain we call the alkyl tail. And because of the photo oxidation, you generate a lot of radicals, and then you add a lot of oxygen. You break the ring open, and become a lot polar. You add a lot of oxygen in, and you generate a very small fraction of CO2. But mostly, this is the compound we see on the photo generated L14 based on this both NMR data and the thermal slicing data. This is average molecule. But it's a mixture. We know the L14 is a complex mixture, but we think based on this data, there are two take-home messages. One, we don't think the aromatic core is that big. It's really small aromatics like this, 2 or 3 ring PAHs, but they got a long chain alkyl tail. So this is certainly bridged. That's a small bridge. And also, as I mentioned, there are the significant fraction of alkane was occluded, so it kind of embedded into this structure. I think it's a key part of the L14. So you can't extract them with organic solvent, but you can release them by pyrolysis, by heat sample. Some of the conclusion I think I learned is really the sunlight play a key role to transform to crude oil. Mostly the aromatic changed to the photo-generated L14 within several days. And certainly there are a lot of reasons why. I'm not going to have time to elaborate, but I think this is kind of totally changed our previous view how the oil being measured. And this kind of give you an idea. Before the deepwater horizon oil spill, we think evaporation certainly always the key. We never thought that the photo oxidation that important. And this after deepwater horizon oil spill, especially during the oil deepwater horizon oils, we know the photo oxidation. You can see kind of the thickness of the line represent how important they are. Right, this become really important. We did not expect before the oil spill. I think I convinced you hope I convince you the thermal slicing technique is really powerful because of the two-dimensional data, and you can have multiple ways to demonstrate the data, including the computing of the energy, the binding energy. We also do some future current ongoing work to using the C14 label, the specific aromatic hydrocarbon, to see exactly what they transform to. This is mostly using the high resolution LCMS, liquid chromatography mass spectrometry. Hopefully we can have a better idea what exactly the chemical form they converted to. We have a lot of, we get a lot of structural information based on the pyrolysis and NMR, but we still need more molecule level information. I think that has to rely on the liquid chromatography mass spectrometry. With that, I think that's all I have. Thank you. If you have any questions.
Thank you, Dr. Liu. We do have a question. Let me just read it.
Yeah.
Can you please tell whether the degradation effect of PAH by sunlight was different for light and dark oil? Whether initial PAH concentration is limiting factor on the degradation kinetics? Meaning, does it accelerate if the initial PAH concentration is high?
Can I see the? Oh, okay.
Yes, it's in the chat box.
Yeah, of course. I think that's a great question. So let me. So I think I can still share the slide. I needed to. So, okay, you can see that now, right? Sorry, I have to go back to the right here. I think that's the question.
Yes, I guess.
Yeah, I mean I think the marine pollution bulletin paper has a detailed each. Well, I think here you go. Here you go. I think this is show you the biodegradation. The black bar essentially show you the biodegradation. For example, the methylated phenanthrene and cervicin, they actually have a lot of faster degradation rate in terms of biodegradation. And also, certainly, this left side is the sunlight. Yes, I think the degradation pH, the composition, they're very different depending on the pH. They're not uniform. In other words, the sunlight effect and the biodegradation are very different. I think the second question, the initial pH concentration will affect the connected. We have a follow-up paper. I think the answer is yes. In this particular experiment we're using the 200 ppm of the crude oil. So it's really a relatively low concentration. So you get probably maximum amount of exposure to the sunlight, I guess. And also the fact that this oil is very light. So they maybe the float on the sea surface, water surface, so they have the maximum light exposure. But if you have a heavier oil, you have a higher concentration, I think the rate will probably decrease. I hope I answer your question. Thank you.
Yes, another question. Please give some recommendation for oil spill analysis of water samples, solvent extraction, or any other SPE approach before PY-GCMS.
Yeah, that's also a good question. Yeah, you can using solid phase extraction. I think that's a lot of standard technique, right? The C18, for example, you can filter your water sample and pass through the SPE. That's probably pretty straightforward. And the other technique I using this separatory funnel. 1 liter, 2 liter, you can add maybe 100 or 200 mil of the DCM, dichloromethane, and do 3 times the extraction. I found that's really work well too. So you separate the 2 phase and you do 3 times. You combine the 3 extract and you roll up down to 1 mil or 2 mil. And then you're ready for your GCMS or GCFID analysis. I think that's also a very easy approach to do. I think the recovery will be better than the SPE, honestly. I think that's probably, probably you just need a little bit of labor intensity. You have to shake separate the funnel for a couple minutes, 5 or 10 minutes or something like that. Yeah.
Okay, and the last question for you is, was there any significant effect on short chain hydrocarbons degradation as well due to sunlight?
Yeah, I mean, if they're aromatic, for sure, because I think it's just the UV, they fit the bonding energy of the aromatic hydrocarbon. Certainly the shorter chain or the last carbon number, they tend to be less photosensitive. So you got the higher number ring you have, I think typically people think is more photosensitive. And you have alkylated pH also even more photosensitive. But if they're straight chain alkane, I don't think there are any light effect. I mean, there may be some indirect. If you have a lot of radicals, they may have a chance of the co-oxidation, for example. But that effect is really small. This question is great. I think it related to people the photogenerated L14. I think there are some folks among the community think there are a lot of saturated hydrocarbon. But the saturated hydrocarbons really come from the alkyl tail of the PAH. So that alkylated PAH being converted to L14. That's where the saturated hydrocarbon from. I think the paper I published, a couple papers demonstrate that really well. So the short answer is they are straight chain saturated hydrocarbon. Usually you don't have the sunlight effect, but you do have the evaporation. I think when you do the experiment, the evaporation, certainly the smaller the molecule tend to be a little bit more volatile. So you have to be careful with that.
Okay. Okay. Thank you, Dr. Liu. That was very insightful. We thank you on behalf of SG Lab Forum, and we look forward to following your future works as well.
Thank you.